Let us now review the electrophoresis of nucleic acids. Electrophoresis is a method of separating nucleic acids based on their size and charge. The process uses a sieve-like matrix, typically agarose or polyacrylamide gel. When an electric field is applied, the nucleic acids migrate through the matrix. The rate of migration is inversely proportional to the logarithm of the molecular size. In other words, smaller DNA fragments move faster than larger ones. Because DNA carries a negative charge, it naturally migrates toward the positive electrode, the anode. Another form is capillary electrophoresis, or CE. This technique uses a thin, fused silica capillary tube instead of a slab gel. Capillary electrophoresis offers high sensitivity, high resolution, and full automation, making it very useful in modern molecular laboratories. A more specialized technique is pulsed field gel electrophoresis, or PFGE. This method is designed for the analysis of very large DNA fragments, up to 100 kilobases in size. PFGE achieves this separation by applying an alternating or pulsed electrical field. This allows the large fragments to reorient and move through the gel matrix more effectively. PFGE is most commonly used for genotyping prokaryotes, especially in epidemiological studies to track outbreaks. In summary, standard electrophoresis separates DNA by size and charge. Capillary electrophoresis provides higher sensitivity and automation. PFGE allows for the separation of extremely large DNA fragments and is widely used in public health microbiology. Let's review hybridization technology and its applications. We begin with the southern blot. This method is used to detect specific DNA sequences. DNA is first denatured in the gel by an alkaline solution. The fragments are then transferred onto a membrane by capillary action using a high-salt solution. A labeled complementary probe is added, which hybridizes to its matching DNA sequence. After washing to remove unbound probe, the DNA is visualized using radioactive isotopes, chemiluminescent dyes, or colorimetric techniques. The steps include restriction enzyme digestion, electrophoresis, denaturation, transfer, probe hybridization, washing, and visualization. The northern blot is similar but detects specific RNA sequences. RNA is transferred to a membrane and hybridized with a labeled complementary probe. The western blot detects specific proteins. Proteins are separated by SDSPAGE, then transferred to a membrane by electrotransfer. The membrane is incubated first with a primary antibody and a blocking solution, then with a secondary antibody. After washes, a substrate is added, and the membrane is developed to visualize the protein bands. The southwestern blot combines features of southern and western methods. It is used to detect DNA binding proteins. Dot and slot blots allow for a quick analysis of DNA or RNA, but do not provide information about fragment size. They are commonly used in expression analysis, mutation analysis, and amplification analysis. Reverse dot blots use allele-specific oligonucleotide probes. These probes hybridize to test alleles and are particularly important for genotyping common human mutations. An important concept is stringency. This refers to the conditions under which hybridization occurs. Increasing salt concentration, heat, or formamide increases the stringency, which controls how specifically probes bind to their targets. Finally, DNA microarrays, also called DNA chips, allow for the simultaneous monitoring of gene expression across thousands of genes. DNA sequences are arranged on a solid support, and probes hybridize to these sequences. DNA microarrays are widely used for gene expression studies, disease diagnosis, and pharmacogenetics, such as drug discovery. Specialized instrumentation is required both to generate the microarrays and to analyze the results. Let's explore FISH, CGH, sequencing, gene cloning, and DNA polymorphisms. We begin with fluorescent in situ hybridization, or FISH. This technique involves the hybridization of a fluorescent DNA probe to its complementary DNA sequence in morphologically preserved tissue or cells. It is used to detect and localize specific DNA sequences or entire chromosomes. FISH can be applied to metaphase chromosome spreads as well as interphase cells, making it useful in both dividing and non-dividing cell studies. Next is comparative genomic hybridization, or CGH. This method compares copy number changes between a test DNA sample and a control. It generates a map of chromosomal regions that are gained or lost, and can also identify the specific location of extra or missing chromosomal material. Moving on to DNA sequencing, which determines the order of nucleotides in a DNA molecule. Several methods exist. Maxim-Gilbert sequencing, based on chemical degradation. Sanger sequencing, also known as dideoxy chain termination. Pyrosequencing, a sequence by synthesis method. Next-generation sequencing, which can sequence single DNA molecules in real time, requires significant computational analysis and is available across various platforms. We then consider gene cloning, also called recombinant DNA technology. This process involves cutting DNA from two different species with restriction enzymes, then ligating it into a vector. The recombinant DNA is introduced into a suitable host cell, which multiplies and produces the desired protein encoded by the inserted DNA. Finally, we examine DNA polymorphisms, which are differences in DNA sequences that occur at various locations in the genome. These include RFLPS, or restriction fragment length polymorphisms, VNTRS, or variable number tandem repeats, also called mini-satellites, STRS, or short tandem repeats, also called microsatellites, SNPS, or single nucleotide polymorphisms, which represent more than 5 million sites in the human genome. Many single nucleotide polymorphisms are associated with disease risk or drug resistance. YSTR haplotypes, which are paternally inherited and used for tracing paternal lineage. Mitochondrial DNA polymorphisms, which are maternally inherited and analyzed in the hypervariable regions for maternal lineage studies. In summary, these tools, FISH, CGH, sequencing, cloning, and polymorphism analysis, are central to modern molecular diagnostics, enabling us to detect, map, and analyze genetic variation with precision.